Hello friends, today we are going to discuss one of the important topic in biomolecule chapter, that is different structures of proteins. Protein structures. That means primary, secondary, tertiary and quaternary. Before that, we have to discuss some of the basic issues of protein for better understanding. Okay, we know that proteins are the high molecular weight, most abundant nitrogen rich substances, and they constitute or occupies 50% of the cellular dry weight. We know that proteins are nothing but polymers of L alpha amino acids. They are nothing but the polymers of L alpha amino acids. L alpha amino acids. We'll discuss point by point, okay? L alpha amino acid. The basic representation stands like this. There are different groups. R group, H group and NH2. This is L alpha amino acid. That means amino group and carboxyl group. It's D form. For example, the same but in reverse. D alpha amino acid. We know that amino acids are the basic bricks in a protein. In nature, there are nearly 300 reported amino acids. But in our protein structure, there are generally 20 amino acids which are considered as the standard amino acids. Out of 300, 20 are considered as a standard amino acid. Standard. Why? Why standard amino acids? Because they are based in genetic code. You know, we have different amino acids like glycine, valine, arginine, phenylalanine, etc. But why only 20? Why not 300? Because of their genetical link. The genetic code, we have 64 codons, out of which 61 are sense codons, but 3 are nonsense codons, and these genetic codes support for the incorporation of 20 amino acids only. That is why they are considered as the standard amino acids. Okay, we'll discuss point one, why they are and what are they, for example. Okay, so genetic code basis of criteria is the judgment for considering a group of or a set of amino acids as standard amino acids. We know that there are different classifications of amino acids. Okay, for example, amino acids based on acidic or basic group, etc. But most conventional is essential or non-essential amino acids. What are essential amino acids? Essential and non-essential amino acids. Non-essential. Why they are some are why some are, that means out of 20, 10 are considered as essential and remaining 10 are non-essential. More specifically, 8 are essential and 2 are semi-essential. Why they are considered as the essential amino acids? Because our biological machinery won't support to synthesize 10, that means essential amino acids, though they have to be supplied through diet. Supplying through diet is most important. Supplying through diet is most important. Therefore, external source we have to depend upon. Without the external source, we cannot get it. But there are non-essential amino acids wherein our biological machinery have enough enzymes to synthesize. And what are the essential amino acids? In simple codes, we can say like A, V, Hill, MPTT. A, V, Hill, MPTT. A stands for arginine, V stands for valine, H stands for histidine, I stands for isoleucine, L stands for leucine, another L stands for lysine, M stands for methionine, P stands for phenylalanine, T is transferred tryptophan and another T tryptophan. So arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan are the essential amino acids. I told you, out of 10, 8 are essential exclusively and 2 are semi-essential. Arginine and histidine are semi-essential amino acids because adult human beings can synthesize. That means we can able to synthesize, but not the children. That is why they are considered as a semi-essential amino acids. So these are about basic issues of amino acids. The rest of the amino acids, that means excluding the essential or non-essential amino acids. Okay. So we already discussed proteins are nothing but a polymers of L amino acids, and it is a basic structure. That means one R group, one H group, COOH group, and NH2 group. That means totally 3 different groups are present. Except glycine amino acid, rest of the amino acids contains these 4 different groups. R groups with respect to means side chain. Okay, side chain. Since 4 different groups, that means R, H, carboxyl and amino group are attached to one carbon, it is considered as an asymmetrical carbon. Asymmetrical carbon. For an asymmetrical carbon, it leads to optical isomers, L form and D form. This is L form, its reverse is called D form. L amino acid, D amino acid and L amino acid. But in our protein structure, L amino acids are present, and the kind of amino acids are called alpha. This is very important. Why they are considered as alpha? Because both functional groups. What are the functional groups of amino acids? Amino groups and carboxyl groups. Amino groups and carboxyl groups both attached to one carbon. That is where they are considered as alpha. Okay, there are different groups, alpha amino acids, L amino acids, and so this is about different structure. Okay, asymmetrical. Why they are considered asymmetrical, and why they are why L and why alpha. So these are the basic terminology we have to learn for better understanding of proteins. Okay, and let us move to the main topic, that is the protein structure. I told you there are different structures. Say primary, secondary, tertiary, quaternary and quaternary. Before we go into that, I will discuss some of the important words. What is the distinction between a polypeptide and a protein? Polypeptide, there are 2 schools of thoughts. Some scientists considered as a polypeptide having minimum of 50 amino acids. That means more than 50 amino acids, they are considered as a protein. That means a polypeptide having more than 50 amino acids is considered as a protein. But another school of thought is there. They say that to categorize or to specify or to depict a polypeptide as a protein, it should have a quaternary structure. That means a polypeptide attaining a quaternary structure may be considered as a protein, or a polypeptide having more than 50 amino acids, it means protein. That means between 10 to 50 amino acids is a polypeptide, more than 50 amino acids is a protein. And then another most important concept before we get into that, okay? Peptide bond formation. Now, better to intervene with primary structure, okay? Okay, first the primary, secondary, tertiary, quaternary, and what exactly the primary means. We told you already. For better illustration, it's simply like primary structure is a linear sequence of amino acids. That's it. It's a primary structure. It's a secondary structure. It's a quaternary structure. Sorry, a tertiary structure. And it's a quaternary structure. That means quaternary structure have more than 2 or more than 2 amino acids or monomers. This is primary, this is secondary, this is tertiary, and this is quaternary. In quaternary structure, they are called as subunits or monomers. Subunits are monomers. Okay, it's a basic representation. We'll discuss point by point. Okay, first, primary structure. Primary structure. It means simply, I told you, linear sequence of amino acids. Linear sequence of amino acids. For illustration, amino acids are the bricks. The primary structure is a wall of a house. Secondary structure, that means the twist. And the third tertiary structure are the rooms, and different rooms in a building constitutes quaternary structure, as a basic analogy for basic understanding. I told you the primary structure is nothing but a linear sequence of amino acids. A linear sequence of amino acids. How they held together. This is very important. Through peptide bond. Peptide bond. And how it will constitute. Okay. Generally, here the diagram depicts the point that it will form between carboxyl and amino groups. Okay, carboxyl and amino groups. Simply, it means that peptide bond will form between carboxyl and amino group. COOH is a carboxyl, amino group NH2. They generally exist in ionic form. CO minus, that means acidic, NH3 plus, basic. By using H2, that means hydrolysis, it forms a peptide bond. This is the peptide bond. So we're clear. Say this is one amino acid, this is another amino acid, and the reacting groups are carboxyl and the amino groups. Hydrolysis reaction, peptide bond formed. That means who are the participant? That means CONH. And this peptide bond is the most strongest bond, most strong enough, and is a polar bond. Because in the next coming structure, that means secondary structures and other structures, it will participate in hydrogen bond formation. That is very important. Peptide bonds are the polar bonds which participates in the hydrogen bondings of different structures, or say the secondary structure, quaternary, and the tertiary structure. So these are considered as the essential fundamental bonds and most strongest bonds in a protein, which considered as a functional, and that means structural and functionalist of a cell or a life event. Okay. So this is how the peptide bond has been formed. Okay. So it's about primary structure. As I told you, it's nothing but a linear sequence of amino acids in primary structure. What constitutes primary structure? Nothing but peptide bonds, and this peptide bond formed between carboxyl and amino groups. Then the secondary structure. Secondary structure is a bit complicated, and it is the fundamental for rest of the structures, or even the entire stability of the structure. For a secondary structure, Pauling and Corey, they have proposed alpha helix and there is beta pleated sheets. The most common structure, secondary structures are alpha helix and beta pleated, whereas alpha helix is the most important structure, and it constitutes or it supports the entire protein structures or the remaining consecutive structures, in terms of advancement. How? We'll see, and how it found. Okay. The name it implied, alpha helix. The name it implied, simply it's a coiled spiral structure. It's a coiled spiral structure. For better representation and for clarity, we are showing some important bonds. Okay, this is NH and this is CO. NH CO, then NH CO. NH this is CO. NH. Likewise, they form okay. This is spiral. Okay. Here the most important bond is hydrogen bond. That means in secondary structure, alpha helix structure, the important bond is hydrogen bond. Actually, individually hydrogen bonds are very weak, but collectively they are very strong bonds. And where they form, between oxygen atom of C terminal and the hydrogen atom of N terminal. This is very important. And for clear cut understanding, say simple like this. This is CO and NH. The green dots represent hydrogen bonds. On an average, there are 3.6 amino acids in each turn, and they travel for a distance of 0.54 nanometers, and their occupancy is 0.15 nanometers. Spacing, what we call spacings, and distance, it is called distance of 0.54 nanometers, whereas spacing of 0.15 nanometers, and each turn 3.6 amino acid. That means in each turn I am saying. Okay, we'll discuss how it will run, how it will end up. So these are all different hydrogen bonds. I told you already, individually they are very weak, but collectively they are very strong. And the name it back, their helical element, it is a right-handed, because right-handed helix is more stronger than left-handed because of its orientation. Okay, and it is densely packed, and here the amino acid sequences are very closely located in such a way that there is a possibility of hydrogen bond between the oxygen of C, that means C terminal, and the hydrogen of N. This is how the issue ends. And these alpha helix, they form spontaneously with least amount of or low energy. This is very important. The most stable structures formed with low energy and spontaneously. Most important point. But there are certain amino acids like proline which will disrupt the secondary structure. That means certain amino acids, they disrupt the secondary structure. Okay, so this is about the secondary structure. Let me one of the secondary structure. That means alpha helix. They are coiled, and each coil contains 3.6 amino acids, and the travel distance of 1.54 nanometers, with spacing of 0.15 nanometers, and the hydrogen bond between the hydrogen oxygen atom of C10 terminal end and the hydrogen atom of N terminal end, and it is right hand helix because of its orientation. Amino acids like proline, they disrupt the alpha helix. Okay, and how about beta helix? Beta helix is very simple. Unlike alpha helix, beta helix is constituted by, say, two segments of polypeptide chains. For example, this is one polypeptide chain for basic illustration, and it is another polypeptide chain. It's also hydrogen bonding only between NH and CO. Between NH and CO. This is NH and OH. This is how the, say, it is beta pleated. When compared to the alpha helix, simply called beta sheets. When compared to the alpha helix, beta plated sheets are somewhat less complicated. In one protein structure, we can expect both alpha helix and beta plated sheets. Okay, this is the second structure. So here the important bonds are called hydrogen bonds. Okay, then the third one, the tertiary structure. Tertiary structures, here I told you already, peptide bonds, I mean the primary structure is a basis, secondary structure is for advancement, and this is over. But tertiary structure is called the mere extensions are based on the requirements. Say for example, there are different bonds involving the tertiary structure. There are different bonds, and most important, and how the tertiary structure is established. Inside the hydrophobic side chains well present, whereas hydrophilic, that means they are present outside or external surface of protein, hydrophilic groups. This is for stability, this is for stability. And different bonds involving in tertiary structure. I told you already. What is a tertiary structure? It is nothing but the three-dimensional element of it. We have already drawn. The three-dimensional arrangement of a protein is secondary structure. And in case of secondary structure, we are well established. That means the spatial arrangement of a protein by twisting. The most important secondary structure is the twisting of polypeptide chains. Whereas in case of tertiary structure, it leads to spatial. It leads to three-dimensional structure. Three-dimensional structure by formation of additional bonds. Here different bonds. Here most important arrangement, what we call most important arrangement, is inside the hydrophobic side chain present, whereas outside hydrophilic groups are present. Apart from hydrogen bonds, we also expect disulfide bonds and electrostatic bonds or ionic bonds, simply hydrophobic bonds. So these are the important bonds in case of tertiary structure. That means in tertiary structure we expect three-dimensional for overall functional activation. Okay. Whereas in quaternary structure, it's about generally most of the proteins constitute of tertiary structure. But in case of some proteins like hemoglobin, we have a quaternary structure. Well, I told you already, there is one school of thought that considered a polypeptide protein when there is a quaternary structure. In quaternary structure, this is nothing but two or more subunits. I told you already, there are different subunits. It is one, and this is another one, and this is another one. For example, here there are three subunits, and they will link together by different bonds. Here, most important, they are mostly non-covalent bonds. For example, hydrogen bonds, hydrophobic bonds, electrostatic bonds, and van der Waals forces. So these are all non-covalent bond. Here the thing is, individually they are considered as monomers, and different subunits of a protein in quaternary structure they are considered as overall oligomers, and individual components are called monomers, and these monomers they attach themselves to non-covalent bonds like hydrogen bonds, hydrophobic bonds, electrostatic bonds, and van der Waals force. So non-covalent bonds plays an important role. For example, in hemoglobin is alpha 1, alpha 2, beta 1 and beta 2. Same for example, you can add one more. This is hemoglobin. That means 4 different monomers constitute a second quaternary structure. So primary structure is based on peptide bonds, secondary structure is based on hydrogen bonds, tertiary structure including with hydrogen bonds, we have other bonds like hydrophobic bonds, electrostatic bonds, whereas in quaternary structure the non-covalent bonds like hydrogen bonds, hydrophobic bonds, electrostatic bonds, and van der Waals forces. So broadly we can categorize proteins, that means the different kinds of bonds, into two types. Based on this we can say simply, it is the last concept, okay, so that we can, for better understanding, different bonds like covalent and non-covalent. In case of covalent, they are peptide bonds and hydrogen bonds and the disulfide bonds, whereas in non-covalent, hydrogen bonds, hydrophobic bonds, electrostatic bonds or ionic bonds, and van der Waals forces. Disulfide bond generally formed by here between, for example, sulfhydryl groups. Say two cysteine residues form cysteine. This is very important. Cysteine. The cysteine becomes cysteine. So different bonds. These are the different bonds involved in protein structure. I hope you understood different issues of protein. Okay, in some other class we'll discuss about some other important biomolecules. Okay, thanks for listening. And I told already, my aim objective is to cater the needs of poor and talented students who cannot afford high lump sum coaching classes. Okay, it may be useful for NEET, CSR, and ICM and other exams. Thanks for watching.